that, I would like to welcome our speaker for today. That would be Daniel Krentzel. Daniel is a doctoral student at the Imaging and Modeling Unit at the Institut Pasteur. He's working on developing deep learning methods for antibacterial drug discovery. But his deep love for deep learning methods goes back to his studies at the Imperial College in London, during which time he also worked at the Francis Crick Institute to develop deep learning tools for volumetric image segmentation and multimodal image registration. And today we hear about the very cool tool called CLEMReg. So please, Daniel, take it away. We are very excited about your talk.
Okay, thanks for this very nice introduction. I think I'm very happy to be here and to talk to you about ClamReg today. Which is, as you said, work that I started while at the Crick. And just to briefly sort of, I mean, you basically preempted this part already from my slide, but essentially ClamReg was something that came out of a sandwich placement that I did at the Crick while I was doing a master's in London. And it's a project that involves many more people than just myself. So it is originally a collaboration between the lab of Lucy Collinson, the MSTP, at the Crick, and Ricardo Henriques, who is now in Portugal. And the main people involved in this project have really been MC Domar and Chris Petty from the MSTP, who have done the image acquisition, the alignment of the CLEM data that we acquired, and also a lot of user testing and given a lot of feedback. The main people involved in the software development have been Martin Jones and Mathur Elphik, together with me. And incidentally, Mathur also did a sandwich placement at the Crick, and he's been contributing great stuff to the project. So let's get into it. Yes, so this talk is going to be about correlative light and electron microscopy, which is oftentimes abbreviated as CLEM. And I thought it'd be a good idea to sort of introduce sort of the concept of CLEM and a bit about electron microscopy as well, for those people who maybe don't know that much about it. And essentially, correlative light and electron microscopy is a kind of a method where you combine fluorescence microscopy with the volumeEM. And the idea is that you sort of combine two modalities that have very different but complementary sort of advantages. So fluorescence microscopy, as you probably know, is compatible with live imaging. You can do functional labeling, so you can label structures in your cells of which you're interested, or proteins and so on. But the resolution compared to EM is much lower. And also you have much less context because you're only labeling what you want to see. So you really don't see anything else but the fluorophores that you added. In contrast, volumeEM has much higher resolution, and you get the entire context of what you're looking at, which in a way can also be a disadvantage because it's a very non-specific type of imaging. And these cells are also very dead, and you will see when I talk about it a bit later that this is a very destructive imaging method. So once you've imaged it in volumeEM, the cell is gone, and you can't really do anything about it. And by combining these two, the idea is really to create something that is more than a sum of its parts, to really take advantage of both of these modalities. And but really, I think visualize this a bit better. Here is sort of a kind of visualization where we have an amphipod, where we zoom into like a diatom, and then we zoom down to a bacterium here. And I think you can appreciate that this bacterium is sort of like half a micron versus sort of, 1 or 2 microns large. And if you were to look at this, for example, in light microscopy with bright field imaging, you would only see something like this. And this, of course, is not very useful if you're trying to look at very small structures. And for example, if you don't look at it in fluorescence, you would have something like this. But of course, if you have this entire image and you wanted to look, for example, at your little bacterium and you didn't know where it was, then it would be quite hard to actually find it. So in this way, if you have fluorescence, for example, you can really sort of find this little needle in the haystack of this EM image. And this is why this is important, and this is why this sort of is a useful imaging modality to look at. So EM comes in various different flavors, and but the kind of core concepts are the same. So essentially you have an electron beam that you use to look at your sample, and you use electrons rather than light because you have much shorter wavelengths of around a nanometer or less. And this means that you can go way beyond the diffraction limit, and you can get images like this one here, which is an SEM image, and I think it's sort of a skin cell, I'm unsure, but I think this is what it is. And the way this works is that you have your electron beam that comes down, it hits the sample, And then you have these like secondary or backscattered electrons that get picked up by the detector. So SEM is really a method to sort of look at the outside of your sample. If you want to look inside of your sample, you would do something like transmission electron microscopy or TEM, where what you need to do, you first sort of embed your cell in resin or in ice, and you then cut a very thin section through it that can be around 70 nanometers or so thick. And you take this little section and then you put it into your TEM microscope, which you then use to sort of look through your sample. And the way this works is it's a bit different from the scanning electron microscope, but the core concept here is that you have your specimen that you put inside of your electron microscope. You again have an electron source with this electron beam that goes through the specimen, and you essentially see the shadow of your specimen here at the bottom. And the images you get look something like this. So you're really looking at the inside now and not at the outside anymore. Now, if you want to sort of get a volumetric perspective on your images, or sort of a 3D image, one way of doing this, for example, is to do serial section TEM, which is sort of, I suppose, the original method of doing this. And what you do is essentially you take this kind of ultramicrotome and you have a diamond knife and you cut your sample on it repeatedly until you get these tiny small sections that you see here. And these small sections you then take and you put them on this very, very small copper grid that is like 3 millimeters wide. And on this little copper grid, you then take this and you then place it again in your TEM to image it. And this way you can build up your 3-dimensional image. Sort of more recent methods that are a bit more advanced, they kind of automate this cutting. Or for example, serial block phase SEM. So this is SBF SEM. And here, what you do is that you have, again, your SEM. So you now sort of look at the surface of the cell. But the way you do this is that you have a diamond knife that will now come in and it will cut through your resin or your sample here. And as you can see, this is a process that is very destructive. So you actually cut off this cell, these like thin sections, and you get very nice images like this on the right here. So this is an image obtained on an SBF SEM microscope. And what you can see is that you can also like have the volume like this, and looks very nice. So if you now want to do CLEM, you of course have to combine these two things together. And I thought I'd illustrate this on a sort of case study from a paper that had the involvement of the EMSTP, where they were looking at sort of trying to differentiate, I think, between tuberculosis bacteria and the host membrane. So the first step here is to really grow your cells on like a little dish here, like this plate, and then you put your cells in a single cell layer on it. And the challenge here is, of course, that you have to find one specific cell that you want to look at. So you identify the cell here on this, for example, this little square here marked with 9K, and then you image it in fluorescence, and you get these kinds of images from it. And in the next step, you then have to actually add the resin, and you have to add these dyes. So you dye your cell then with these heavy metals so that you can actually get some sort of contrast in your EM. And once you have done this, you sort of also have to dehydrate it, sort of get out all the water and so on. You have to then cut this little piece that you have found, this sort of this little square where you know that you have your cell. You cut this to shape in like this pyramidal shape that you can see on the right here. And then in the next step, you have to actually cut off this top bit with this razor blade. And we're talking about like a little thing that is sort of 500 microns wide and 2 millimeters tall. So it is really something that is for people who, you know, don't drink coffee in the morning and kind of are very good with sort of very dexterous with their hands. And you see, so you cut off this piece here, and then you have to actually mount it in your electron microscope. So in this case, we're doing serial block phase SEM. This is this method where you cut through your sample and then have your scanning electron microscope that looks at these different sections that you produced. And you can see here where you have this little cocktail pin, which is again sort of a millimeter big, like it's tiny scales we're talking about here. So you take this little block and you glue it onto this cocktail stick, and then you have to take it and then mount it on here so that you can then put it inside of your electron microscope. So we're really dealing with the micro scale here. And yes, so this is what it then looks like. And you mount it here, and then you start with the imaging. And once you have done these two steps, you end up with these two these two different images that on the one hand, you have the functional information on the left that you get from the fluorescence microscopy. So in the top, what you have is something that stains the mycobacteria. And on the bottom, you have a stain that sort of highlights where the host membrane is, because the goal is to sort of differentiate between the host membrane and the mycobacterium. And on the right here, you have the entire structural context of your cell. And I think you can sort of see a hint of a shadow in here that is this is sort of corresponds to this image here, roughly. So then what you can do, you can overlay these. And when you have this overlay, you can then really see the power of CLEM, where you can really sort of differentiate certain bits in your EM image from other bits, of which you don't know what they are if you only had the EM image. So this is the advantage of this. But the bummer with this is that intensity-based registrations don't really work here. And this is, in a sense, more, I guess, a feature than a bug, because you really do want to have different information from these two imaging modalities. Otherwise, you wouldn't go through this kind of process. But it means that currently the state of the art in aligning VCLEM datasets is really having somebody who sits down and who identifies landmarks in these two different volumes. So you have somebody who sits down and then sort of looks at these two image stacks and says, okay, this point here looks like it could correspond to this point over here. And then you repeat, and you do this for various different points until you have a list of landmarks that, that essentially allow you to define a transformation from your fluorescence image, which essentially is a set of points that you now have, and you then work these points onto your target, which is the EM. And once you have found this kind of transformation, you can apply it to your fluorescence data. Now, like everything with EM, this is also very hard to do because you're working with like sub-micron alignment here of quite large image volumes sometimes. As you saw previously, the sample processing can really introduce quite large deformations because you sort of mechanically cut your sample. You're also imaging it in two different types of microscopes. So it means that you have to mount it in one microscope, but you viewed it in a different orientation in the previous microscope. So these are all challenges to consider. And also working in 3 dimensions is quite hard. So it's quite hard to sort of wrap your head around sometimes what corresponds to what when you have sort of obliquely lying objects. And also the placement of these landmarks is subjective, and you usually need quite highly trained people to identify these landmarks. And it's quite time consuming as well. And also annoyingly, the more points you add, it seems like it doesn't really get better. So your alignments a lot of times sort of you max out quite quickly, and then adding more points a lot of times actually does not improve the registration, at least in the manual case. So the obvious question for me at least is, okay, has anybody tried to automate this before in some way? And the answer is yes. There has been prior work on automating CLEM alignment. And there are sort of 2 different flavors to this. So on the one hand side, people sort of try to look at converting in some way the EM data or the fluorescence data to look more like one of the other modalities. So for example, up here, what the authors did is that they took the bright field data, they processed it, and they tried to make it look as similar as possible to the EM data, and then try to align it that way. Here you have a deep learning based method, which came out quite recently, and it's like a nice paper where what they do is that they take the electron microscopy image and they try to predict a fluorescence image from that, and then they use those images to actually align them. The issue with deep learning, of course, in this context is that you need to have a lot of data to actually do this very well, and it's hard to generalize across different types of modalities in EM sometimes. So this works only in 2D as well, but this is oftentimes sufficient for a lot of tasks. And in these last 2 papers here, what they do is that they actually introduce changes to the overall protocol, or they require sort of additional hardware for this to work. So what we wanted to do is to have a pipeline that does the registration sort of natively and fully in 3 dimensions. We didn't want to have any changes in the hardware or in the protocol, and we wanted to make it open source and easily accessible for end users, because we thought that was quite important. And also we openly share all the benchmark data that we have acquired. So they've been uploaded to Empire, so that other people can sort of go ahead and have a look at seeing if they can also develop their own registration algorithms, for example. So for this project, the first step, of course, is that we need some sort of benchmark data that we can use to develop this method. And here we had Chris and MC who were super important for this. So they acquired 3 different benchmark data sets, 2 in FibSem, and one data set in serial block phase SEM. And they labeled these stacks with 4 different dyes. So we labeled Mitotracker, Lysotracker, TJ46, which largely labels the Golgi, and then Höchst. And then we have these FIB-SEM stacks. And once that was done, MC sat down and did the manual alignment of all these data sets using Big Warp, which is a software that is widely used in the community because it allows you to sort of really easily kind of scroll through these large stacks that you have and place landmarks and also compute the transformation and apply it to the fluorescence image. So once we had these data sets, we had to think a bit about how to go about actually aligning these data sets and what the strategy would be here. And we thought a bit about sort of what people do in the sort of manual case, where what they do is that they try to identify structures that are sort of shared in both modalities. And we were thinking if we could find some structure that you could automatically identify in both of them, and that is abundant and sort of a bit everywhere in a cell so that you get a good alignment. And we quite quickly settled on using mitochondria because the limiting factor here is really not sort of segmenting or identifying structures in fluorescence because you know what you're tagging, right? I mean, if you, for example, tag mitochondria, then you know this is the signal that you get. Whereas in EM, because you have the entire ultrastructural context, you sort of need to have a bit more advanced methods to actually identify the different objects in there. And what was quite important here was to have this Mito-EM challenge, which was a challenge that took place in the context of ISB 2021, where essentially teams were invited to submit their own mitochondria instance segmentation models, and they then compared them and benchmarked them, and this really drove forward the development of new models. And just as an example of what this looks like, so here is the mitochondria segmentation that was submitted from the CRIC, which also involved the EMSTP. And I think what is very clear here is that the mitochondria are really abundant throughout the cell. And what is more important is that now it's possible to automatically segment them. So it's pretty much a solved problem. The next step was then to find the mapping between the fluorescence and the EM. And then lastly, we needed to find a way to warp the fluorescence onto the EM. So I'll talk through all of these points. So the first step, of course, was segmenting the mitochondria in fluorescence. And so what we did is that we simply used Mitotracker to identify the mitochondria in the fluorescence. And the task here is more sort of trying to separate the foreground from the background, or the signal from the noise, really. So we did some pre-processing, which involved sort of interpolating these stacks because the Z resolution is much worse than in X and Y. And we then applied a Laplacian Gaussian filter because we found that worked the best at really separating between the mitochondria and the background. I think you can see that this works quite well here. Next, what we did is that we just applied a size-based filter to our segmentation that we have to kind of get rid of spurious small objects that we're not interested in. And then you end up with a 3D segmentation that looks like this here on the right. And here are some overlays from this. And I think you can see that in blue or cyan, you have the Mitotracker. And then here in red, you have the segmentation masks that you have. And I think it's quite clear that we're able to sort of separate or sort of identify the mitochondria quite well in these stacks. So we were happy with that. Then the next step was to segment the VEM data. And we tried different methods to do this, but we found that the one that worked the best was MitoNet, which I think also came out of the Mito-EM challenge. And it's a pre-trained model that has been trained on a large variety of different types of EM data. And it generates beautiful segmentations like the ones that you can see here on the right. So this really works very well, and it worked across different types that we also tried ourselves. So we chose to use this as our way of segmenting the mitochondria in EM. And here you can see the overlays that you get from the segmentation in EM. And again, you can see that these are quite neat, and we were also happy with these. The next step is then trying to sort of extract landmarks in a way from these segmentations. And the way we did this is that we simply sampled point clouds on these segmentations. So what you can see is that essentially we identify the surface of them, and then we place points randomly on the surface and then downsample it a bit so that we can actually reduce the complexity or the sort of computational load of then doing the alignment later on. And one could ask why point clouds and not sort of directly using the segmentations. And the reason is that point clouds are inherently sparse. So it's a three-dimensional representation, which suits our needs. And because it's sparse, it's also inherently much more memory efficient, because you don't have to store a value at each voxel that you have, but instead you just store the sort of the Cartesian coordinates that you have inside of like a list, essentially. And also these point clouds are, of course, modality agnostic. So it doesn't really matter where they came from. It's just that now they're two clouds and you can sort of do stuff with them. And what is nice is that point clouds are used widely in other domains. So for example, 3D scanners use them, or for example, people use them to scan buildings, or in robotics or self-driving cars. So it's a very active research field. And it means that we can sort of reuse algorithms that are robust, but that have been developed in other domains in this context. And so the sort of go-to approach that people typically use when they do point cloud registration is that they use iterative closest point. And the issue here is that essentially in these point clouds, what is different from the landmarks that we get manually is that in the landmarks, you have a direct correspondence, like you know what point corresponds to the other point. So you can use sort of simpler methods to just like find a warping there. Whereas here, you don't have these direct correspondences. So part of what these registration algorithms need to do, at least in this case here, they need to establish which points belong together. And this is what iterative closest point does. And it essentially estimates the point correspondence between some sort of initial point cloud, this like moving point cloud, and a target point cloud by simply matching the closest points together and then sort of updating such that this distance minimizes constantly. And so there's like an iterative process that takes place. And in principle, that sounds good, but that did not work for us at all. So here on the right, you can see sort of the result of using iterative closest point. And the issue with ICP is that it usually ends up on sort of local minima, and it becomes an issue if you have like these dense points here, like in the segmentation. So we had to think a bit about sort of what was going on with our point clouds and why this didn't work. And one of the issues, of course, is that you sort of have these random points that we have sampled, so you don't have this like direct correspondence. So we needed to think a bit about this in a different way. And there's a different method that is called coherent point drift. Then instead of looking at this as a point-to-point correspondence problem, it actually casts this registration problem as a probability density estimation, where essentially one of the point clouds is some sort of mixture model, I think a Gaussian mixture model. And then the idea is to try to maximize the likelihood that one of the point clouds sort of was sampled from this first point cloud. And this way you sort of become much more robust to missegmentations and outliers. And this is quite nice for this case here, because it is, as you can see, these point clouds do not look exactly the same. They look similar, but they're not exactly the same. And the other very nice thing here is that coherent point drift also has implementations that are nonlinear. So for example, Bayesian coherent point drift is a version that is nonlinear, so it allows you to do this nonlinear mapping, which is quite nice. And if we use coherent point drift, this is what the result looks like. So you can see that this works much better than the ICP approach that we tried first. And what you can also look at is sort of the overlay after we have these converged point clouds. And here in red again, we have the point cloud from the moving cells, and then in blue we have the target. And once you find this alignment, either be it through rigid registration or with nonlinear registration, you now have established essentially a point-to-point correspondence where you know how to map from your fluorescence microscopy image to your electron microscopy image. And you can then apply either your transformation matrix in the case of a rigid alignment, or we also included a thin plate spline warping so that you can sort of use these points essentially as control points to do this nonlinear warping. And if you do this, you can then apply this to all the channels, not only the mitochondria channel, and you end up with something like this, where you have the mitotracker in cyan, you have the lysotracker here, TGN46 and Höchst, and this is automatically registered with ClamReg. And I think you see that it sort of works pretty decently. at least qualitatively. And we then applied this as well to other stacks. So on the left here, we have another FibSem stack that we had acquired. And on the right here, we have an SBF SEM stack that is also automatically overlaid. So if you take a look at this sort of qualitatively, what we have, so here on the top, we have sort of the CLEMReg, the automatically aligned image stacks. And on the bottom, we have the manually aligned image stacks. And what you can see is that, at least qualitatively, this looks very nice. And it's sort of, and it seems to work well. And you can also take a look then, for example, at the other FibSem stack that we aligned. And again, the story is quite similar to the previous image, is that the sort of overlays look very, very similar. But the question then, of course, is can we quantify this in some way? So can we quantify the alignment quality? Because simply looking at these images and saying that it looks nice is not necessarily the most satisfactory way of assessing your alignment performance. So we thought about this a bit because this is not so straightforward. And it has to do with the fact that, I mean, again, just wanted to stress this point that there is really no way of measuring the agreement between your electron microscopy image and your fluorescence microscopy image in terms of any intensity-based measure, right? So this is so this is not possible to do. So we had to think a bit about sort of, you know, do we want to compare it to the manual alignment that we have? But then there again, you know, the manual alignment is also not perfect and is also a bit subjective sometimes. So we wanted to have a more objective way of actually assessing this performance. And if you think about the aim of CLEM, it is really to sort of assess the identity of an object in EM using light microscopy. So it's really this idea of like assigning some function to a structure that you have in EM. So for example, if you have the structure here on the right, this might be some sort of EM blob that you have. And then you have sort of your fluorescence blob on top of it. And as you can see, sort of the idea is that you really want to sort of find this kind of agreement here. So we thought maybe what we could do is to try to measure the degree of overlap on sort of some small and sparse structures that are located throughout the image volume. So we decided to look at lysosomes and endosomes because those are quite small and they're a bit everywhere. And to then actually measure this overlap between the EM and the fluorescence, we decided to use the true positive rate instead of sort of more commonly used metrics like the intersectional reunion, which is oftentimes also called the Jacquard index. And I will motivate this in a bit why we do this. But essentially, the true positive rate, what it does is that it computes the fraction of your EM object within the LM signal, which is essentially what we're interested in. And the reason this is a sensible thing to do is that if you look at your EM blob that is here on the right, this sort of represents kind of the real size that you have of this object. And if you now image the same object in fluorescence, what you end up with is sort of this diffraction limited spot that will look much larger because you're using light instead of electrons to image it. So if you now sort of get a segmentation of these two to somehow measure the overlap, and you do the registration between these two, you will, in the case of using intersectional reunion, you will end up with something like this, where what you will notice is that even if we had a perfect overlap, we will never get the maximum score because we will always divide also by the green area, right? So what we're more interested in is actually just looking at sort of how much of our EM object is actually within the signal, within the LM signal. And this is what the true positive rate gives us, which is essentially just looking at the fraction that is correctly inside of our green blob. And so how we did this in practice is that we asked MC to segment lysosomes in the EM volumes that we have. So this is one of the benchmark data sets that we used. And she sat down and she sort of looked and sort of found different lysosomes throughout this volume. And then she segmented them. And what you can see here are these tiny sort of lysosomes segmented. And so this one is the one that comes from here and so on. And what they also show is this in magenta is a lyso tracker, which is the sort of fluorescence signal that we get after doing the ClamReg alignment. So we do the alignment with the mitochondria channel, and then we apply the same registration parameters to our lyso tracker channel, and this is what we get in terms of the overlay. And you can see that all of these objects are sort of kind of correlated with some sort of fluorescent signal. And what we did is that we went ahead and we quantified throughout the volume on these 5 different lysosomes that we looked at. We quantified this true positive rate. And what you can see is that the manual alignment in this case is definitely better than the ClamReg alignment. But what we also see is that ClamReg is actually able to sort of identify all of the, or like assign the signal to all of these lysosomes correctly, which is nice. And then we went ahead and did the same thing in the FibSem, in the other FibSem data set. And here we didn't have a lysosome channel, so we used the TGN46 channel instead, because it also labels endosomes partially. And here we segmented 4 of these endosomes, and we then again computed this true positive rate. And in this case, we were doing much better as compared to the previous cell. And then lastly, what we did is that we took a look at the serial block phase SEM data, which is a different modality from this FibSem. So it's a different way of imaging the EM. And here again, we looked at lysosomes. And what you can see here is more that we have this kind of bimodal situation where we have sort of these 2 top points that perform really well in line with the manual alignment. And then we have 2 points that sort of don't work as well. So we asked ourselves, okay, so what is the reason behind this? Why does this happen? And so we sort of took a look at where these lysosomes are located within our EM volume. So what I'm showing you here are the mitochondria EM segmentations in gray. And then we have the lysotracker signal overlaid to this. And then here we have small lysosomes. So essentially, if we zoom in, this is what we have. So we have these lysosomes and then the signal on top here. And what you will notice if you take this image and you project it down to get sort of a density map of mitochondria, this is a sum projection simply of the EM segmentations, and essentially gives you an idea of where you have more and less mitochondria. What you can see is that the closer you essentially are to the center of the cell and the closer you are to more mitochondria, you get a better performance in your alignment. Whereas the ones that are really at the edge, or here this one, which is completely without any mitochondria around it, and these cells or these lysosomes have a much worse overlay, which sort of intuitively makes sense because we're using the mitochondria for the overlay. So this is really one of the limitations to keep in mind, is that when doing this automated alignment, you really need to have your structure to use for the alignment actually present around whatever object you want to look at. And we then decided to also make this available. So I said that we wanted to make this easily accessible for end users. And we decided to go for napari in this case, instead of using Fiji, for example, which I think is more commonly used in the community. And the reason is really sort of, there are various reasons for this. And the first most important one, of course, is that napari is written fully in Python. So it means that any sort of deep learning pipelines or like deep learning tools that you want to integrate is very straightforward, even though you have solutions in ImageJ, but those are more tricky. And also what is quite important with napari is that you have this 3-dimensional viewer that sort of natively handles 3-dimensional data, which is quite useful if you're doing volume CLEM, which is inherently 3-dimensional. And also you have the option of adding things like points layers or shapes layers and so on, and interact with them directly on the image, which is something that we also do within the plugin that we developed. So installing napari maybe is not as straightforward as installing Fiji, but it's not super scary either. So it's just a couple of lines of code. And I made a very quick video to show sort of that this is really just 3 lines of code. And then to open napari, you just put that into the terminal, and then you open it up. So it's not so difficult. And then what we also did then is that we built a plugin that we put on the Napari hub, which is sort of this place where all of these Napari plugins are hosted, and there are like more and more plugins being uploaded continuously to this site, and we put Napari ClamReg on here. And to install Napari ClamReg, it's quite easy. So again, you just have to go to the install plugins window, and then just scroll down until you find Napari ClamReg, install it, and then restart Napari, and then it pops up if you go to the plugins window. Yes, exactly. And And our plugin, the idea behind this plugin was to really make this as simple as possible to use. So we wanted to have a solution where you load or you choose your image layers that you have loaded into the napari viewer, and we then automatically read things like the pixel size from the metadata, and then we can pre-process the fluorescence data, for example, automatically. And then all you have to do, if the parameters work that we have chosen for you, is just to hit register, and then you get the registration out of there. But of course, you can also choose to kind of change these parameters depending on things that don't work. So for example, you can change the segmentation parameters. So here, for example, you can choose to either do the MitoNet segmentation in just one plane, or you can do it across like 3 axes. For example, it does it across all 3 and averages it. You can change the parameters for the fluorescence microscopy segmentation, and then also choose how many points you want to sample. And this is really, this registration parameter section really allows you to tweak how much memory you use when doing this alignment, because really the bottleneck is the number of points that you have. And then at the end, if you choose to do non-linear warping, because you can choose the registration algorithm that you want to use, either rigid or non-linear, you can also change or choose to chunk your data so that you are really able to warp larger volumes on a smaller RAM machine. And we also have a split registration workflow, which means that you can also run these individual steps by themselves, so that you can do this optimization actually rather than having to run the entire pipeline and then changing parameters every time and then waiting. So this speeds up your optimization for changing these parameters. And also what it allows you to do is that you can essentially choose to use any type of image segmentation approach that you want. So it integrates nicely, for example, with other napari plugins. So for example, this is the napari PyClass Brunt Assistant, for example, and here you can create much more sophisticated segmentation pipelines. And then you can segment your fluorescence image using this plugin, for example, and then just use that segmentation layer that you have directly with napari ClamReg, because you can then, for example, sample your point clouds from an already existing segmentation rather than having to go through the entire process. And also in terms of if you want to change the EM segmentations, you can do this as well. So for example, you can go to bioimage model zoo, where you have various different types of deep learning models, and a lot of them trained on different organelles in EM. And this allows you to also go beyond only using mitochondria. If you, for example, want to look at some other structure, or you have cells that have no mitochondria, or for example, one use case might be that you have a tissue, for example, and you have a bunch of nuclei stained, and then you would instead want to segment the nuclei and then do the registration using those rather than the mitochondrial networks. We also ship CLEMReg with sample data. So you can open your sample data through this files window, and then just load it into the viewer, and then you get this. So if you're interested in using it, you can download napari ClamReg and then just directly play with the data that we provide you with. And you can also choose then, for example, the registration directions. You can choose if you want to go from the fluorescence to the EM or from the EM to the fluorescence. So yeah, and here at the end, I wanted to include also a little demo video, because I was advised against doing a live demo. So yeah. So here what you do is that you load your images in, and you have to open it with the napari ClamReg reader so that we can read the metadata. Then you link these layers up so that they all get processed together, so they all get registered at the same time. You can then load your EM data in, and then just open the plugin. And one thing we allow you to do as well is to choose a region of interest that you want to overlay, so that you improve the performance in certain cases where you might have a lot of other stuff around that you necessarily don't want to have in the point clouds that you generate. So you can mask this a bit, and you can adjust the fluorescence channel. And then you can save your parameters as well that you have chosen, which is quite useful if you want to do batch processing, or you have a situation where maybe you want to write a paper and you need to remember what the parameters were used for a certain registration. And we visualize the intermediate layers as the registration goes on. And So we output points layers, we output labels layers, which again highlights why napari is useful because it handles all these things natively. And here are the point clouds that you get. So in blue, we have the EM point cloud, and in red, we have the fluorescence point cloud. And here's the overlay of these. And then it's done. So as here, you have the overlay of the fluorescence channels onto the EM. And you can see the lysosome channel, for example, in magenta. And then once that is done, you can save it all into a folder using napari. And yes, and that's pretty much it. So everything I've talked about here, we also wrote up as a little preprint that you can find on bioRxiv. And I again wanted to thank everybody who's been involved in this. It's been a great team effort. And